Hello, everyone. Today, we are pleased to present our work on inflammation, immunity, and cardiovascular diseases, published in Med Research. This study was led by Naval Medical University together with collaborators from Gansu University of Chinese Medicine, Tongji University, and other institutions. Our goal is to provide a more integrated view of cardiovascular disease and to highlight its translational value for precision medicine. The key contribution of this work is the IICM framework: inflammation, immunity, cell death, and immunometabolism. This framework connects inflammatory mediator networks, immune cell programs, regulated cell death, and metabolic regulation into one unified architecture. It suggests that cardiovascular disease is driven not by a single pathway, but by a dynamic network state that changes with biological context and disease stage. This slide focuses on the inflammatory response as the starting point of cardiovascular pathology. Chemokines such as CCL2 and CX3CL1 recruit monocytes to the vascular wall, where they differentiate into macrophages. M1 macrophages and Th1/Th17 cells amplify inflammation, while M2 macrophages and Tregs promote repair, and their balance determines disease outcome. This slide highlights the Treg-DC axis as a key immune regulatory module in cardiovascular disease. Activated dendritic cells drive pathogenic T cell responses and lesion progression, while tolerogenic dendritic cells expand Tregs through pathways such as IL-27 and EDOR. This also shows that immune regulation is highly dependent on the disease microenvironment. This slide shows that programmed cell death is not simply the end of tissue injury, but an active part of disease regulation. Apoptosis contributes to cardiomyocyte loss. Autophagy has a dual role in protection and injury, and ferroptosis promotes plaque instability and ischemia reperfusion damage. These different modes provide multiple targets for precision therapy. This slide focuses on immunometabolic reprogramming as a driver of immune cell behavior. A pro-inflammatory phenotype is mainly supported by glycolysis, while a reparative phenotype relies on fatty acid oxidation and oxidative phosphorylation. In other words, metabolic state directly shapes immune function and disease progression. This slide summarizes the feed-forward loop linking inflammation, immune regulation, cell death, and metabolism. Core inflammatory factors such as IL-1 beta, IL-6, TNF-alpha, and IFN-gamma, together with pathways including NF-kappa B, JAK-STAT, and NLRP3, drive a self-amplifying cycle. This loop promotes endothelial dysfunction, sustained immune recruitment, and multiple cardiovascular phenotypes. This slide highlights the clinical value of inflammatory biomarkers. Markers such as HSCRP and IL-6 support broad risk prediction, while LP-PLA2 and MPO help assess plaque instability, and PAGLN and LP add further prognostic value. Multi-marker panels may enable more accurate and stage-specific cardiovascular intervention. This slide focuses on therapeutic translation. Canakinumab, ziltivekimab, and colchicine have already shown encouraging clinical value in cardiovascular inflammation control. At the same time, emerging targets such as chemokine axes, macrophage polarization, immune checkpoints, and immunometabolic pathways are opening new directions for next-generation therapy. To conclude, this study delivers 4 key messages. First, cardiovascular disease is driven by a dynamic IICM network, rather than isolated linear pathways. Second, the balance between pro-inflammatory and reparative immunity plays a decisive role in disease outcome. Third, inflammatory biomarkers are essential for precise risk stratification and personalized intervention. And finally, the future of treatment lies in phenotype-specific, network-level immunomodulation.